Everyone, I found a cool video that introduces genetics, and basically we're going to watch it together. I'll pause it if I ever feel like I need to tell you something. Here we go.
A few years ago, a wave of vitamin A deficiency hit many parts of the globe. The deficiency was so severe that it resulted in several deaths within no time. What could have been done to crack this problem? Scientists soon realized that the staple food of most of the areas hit by this disaster was rice. They took efforts to modify rice in such a way that
Okay, so this begins with a story, and this story is about vitamin A deficiency. So a deficiency means that there is not enough of this vitamin A in these people's diets. And so they were dying, right? And to fix that problem, what they did was they changed rice to have more vitamin A.
it produced the necessary compound in the body when consumed. With this, the concept of golden rice emerged. It's a variety of rice that's capable of biosynthesizing beta carotene, which is a
Let's look at that word, because you might not know what biosynthesis means. It means a living thing is making something. Synthesis means to make, bio means life. Biosynthesis means that you can make it yourself. And so with golden rice, it had the necessary compounds in it to help biosynthesize beta carotene, which is a precursor to vitamin A. Precursor means that it is something needed to make something else. So for example, a precursor to making chocolate milk is milk and chocolate. It's like an ingredient. So this stuff here, this rice, had beta carotene in it that was a precursor to vitamin A.
precursor of vitamin A. When consumed, the beta carotene is readily converted to vitamin A in the body, thus saving uncountable lives. Doesn't this sound like science fiction? It definitely does, but this has actually happened.
So when she says this sounds like science fiction, it is because for a long time we could not do this. But we have learned how to change the genetic information of a plant to make it do what we want it to do. And that's what we did. We put in a special gene. So we put in some sort of gene, and that gene made us the beta carotene.
Let me make it more interesting. Imagine there's one single plant that has potatoes in the root and tomatoes at the shoot. Do you think this is possible? Of course it is. This variety is called pomato, a plant that bears two edible parts in the same body. And now for the last one. Imagine you're in a dark room and suddenly you find a fluorescently glowing mice running around. Do you think this kind of glowing mice exists?
So fluorescent means to glow. And so the way that this works, it's called trans. Trans means to go through or opposite sides. Transgenic. Genic, I think that's how you spell it. So transgenic means to come from another organism. So the organism that actually they stole this glowing gene from was a glowing jellyfish. It has a gene called GFP, and they put that gene into the mouse, which makes it glow. We can do this with cats too. I invite you to pause the video and go Google that. There's glow-in-the-dark cats. It's a real thing. We could do this with you if we wanted to. We could take this jellyfish gene and put it in you, and you would glow. I don't know why you'd want to do that. That'd be kind of hard to sleep, but there you go.
Yes, they do. The concept of fluorescent mice is no longer fiction because there are transgenic mice which glow with fluorescence. Why are we looking at these examples? We know that computers run on codes, and so do our bodies. Just like there are specific codes for running a computer, we have specific codes for organisms' body design. And any change in these codes can give us miraculous results like the golden rice, the tomato plant, and the glowing mice. And the study of all these wonderful concepts is nothing but genetics. We have come across this term several times. Genetics finds applications not only in modern day research, but even answers various queries that pop up in our minds. Many of us assume that genetics is only about the characters passed on to the children from their parents. But is it as simple as that? And even if we know or assume things, it is important to ask the question, why? For instance, have you ever wondered why an apple tree only gives rise to another apple tree? Why don't we get a banana or a mango tree from it? Why does a cat give birth to only a kitten? And why do humans give birth to humans only? Well, you may say that the answer is nature. Okay, so if a human gives birth to another human, then why are those two not exactly the same? Why do we find each individual unique? And what makes the kid strikingly different from his or her parents?
So, basically, you'll learn later that each parent is giving half of their DNA to their child. But it's a little bit more complicated than that, actually. And so instead of thinking it as DNA, I like to think of it as a box of cards. So this is a deck of cards, and this is a deck of cards. They're DNA. And yes, you are giving DNA from each parent, but you're also shuffling that DNA. So it is random what cards you get from the mom and what cards you get from the dad, but then you're also shuffling them together in a very random way. And so we're going to learn what that means later.
Well, there are endless questions that pop up in our minds. And how do we find answers to them? That's where the branch of genetics comes into play. In this lesson and in the upcoming ones, let's get introduced to this branch and try answering all the questions that pop up in our minds about the living world around us. To begin with, let's define genetics. In simple terms, it can be defined as the branch of biology that deals with heredity and genetic variation. Now you may wonder, what do these two terms mean? To begin our studies on genetics, we need to first understand a few terms in detail. Let's begin with understanding what heredity is. Here's one happy family. Now, if we observe this kid, he has a few features like his dad and a few like his mom. That is because he has got these features from each parent. This passing of traits or characters from one generation to the other is nothing but heredity. So we can define heredity as passing of traits from parents to their offspring.
I want to point out that they're only showing physical traits, but there's also behavioral traits that are passed on. You might be familiar with this. Maybe you know somebody. They say that the apple doesn't fall far from the tree. That's an old saying that basically means that the child behaves just like the parents. And so it's not just physical traits that are a part of heredity. It's also personalities and behaviors.
Now, what do we exactly mean by traits and characters? It's simple. In this case, the child has black-colored eyes just like his mom. The father, however, has blue-colored eyes. Now here we have two different types of eye colors, black and blue. Similarly, there are many other colors of eyes that exist in nature. So the eye color is the character. The eye color can vary among the population of a particular species. On the other hand, the trait will be black or blue color of the eyes, which is exclusive for that particular individual. To take another example,
we actually define this better later. So when we talk about DNA, the character is controlled by what's called a gene. So a gene is just a portion of DNA that codes for a protein. So for example, the protein in this woman's eyes that makes them the color they are is melanin. Blue-eyed people have less melanin than brown-eyed people. And so this gene, when it is read by the cell, makes melanin, which makes her eye color. And so the character is a gene. The trait is the particular gene, and we call that an allele. So the better term is allele. You can think of it like this. If a gene is ice cream, the flavors are alleles. Or in this case, if the gene is eye color, the alleles are blue, black, or green.
height is a character, while tall or short are the traits. Hair color is a character, while black and blonde are the traits. The passing of traits from one generation to the other is called heredity. And what has been passed to the next generation is usually referred to as inheritance. Now tell me one thing. If this boy has inherited the traits from his parents, why is he not the exact copy of either of the parent? I mean, why does he look slightly different from both? The answer lies in the single term called variation. Isn't the term self-explanatory? As the name says, it means differences. So variations are the differences that make one organism different from its parents. As in the case, the boy has inherited a few traits from his mum and a few from his dad. But he is different from both. Thus, there are many variations in him. These basics in genetics are known to us now. But ever wondered who laid the foundations of this interesting subject? Who were the pioneers in genetics that helped us crack the code of life? Let's have a glance at the history of genetics in the upcoming part. This image of evolution is not new to us. But ever wondered how species evolved? How are a few factors eliminated and a few carried forward? Well, the answers are obtained when we combine evolutionary studies with genetics. Many scientists have been working on genetics since ages. But there was this one legendary figure who could answer why a green pea plant gave rise to another similar green pea plant. Let's get back in time to understand how the foundation of genetics were laid. Genetic studies were carried out since the classical era, wherein scientists like Aristotle and Hippocrates have put forth a few theories regarding how parental characters are passed to the next generation. However, a breakthrough was achieved when an Augustinian friar carried out remarkable experiments using garden pea plants and laid the foundations of modern-day genetics. It was around the 19th century that Gregor Johann Mendel, now known as the father of genetics, performed the experiments that led him to this path. Mendel, while working in a garden, wondered why the green pea plants with one particular trait gave rise to plants with the same trait. For example, plants with purple flowers gave rise to plants with purple flowers only. So why is this so? And why do white color flowered plants give plants with white color flowers only? And what will happen if these two varieties are crossed?
So the question is, because this is important, that's why I'm going over it. The question is, if purple flowers are expected to only make purple flowers, and white flowers are expected to only make white flowers, what would happen if you made a purple flower and a white flower have sex? When they mix their genetic information together, what do you get? And so a lot of people thought that this would make a pink flower a mix, but that is just not true.
All these queries in mind, Mendel decided to carry out a few experiments that would help him find out all the answers. But where did he think all these experiments would take him? How did he think of beginning with the procedures? And what were the list of requirements he gathered? What could have been the possible conclusions? Let's try answering all these questions one by one. To begin with, let's understand why did Gregor Mendel choose Pisum sativum, commonly known as the green pea plant?
In science, we have two-part names for something. So this particular plant has a genus name and a species name. And then there's the common name. So the common name that most people call this is a green pea plant, but the scientific name is Pisum sativum. And we always capitalize the genus, and then the species name is always a lowercase, and both are italicized.
Mendel knew that for his experiments, he would require a plant which is perennial and not annual. In literal terms, perennial means something that can last for a long time. Here it meant the plant that can grow and reproduce in a recurring fashion. Secondly, the plant should have a small life cycle. It should be able to grow quickly. Also, Mendel wanted a plant that would not require too many exclusive conditions to grow. Minimal requirements should suffice the growth of the plant. Most importantly,
So Mendel actually tried to use other specimens to test this crossing. But he couldn't get them to work. He tried bees, he tried mice, and then he tried pea plants. The problem with animals is they move too quickly and they are harder to take care of. But these pea plants do not move, and they are easy to take care of.
Understand the results of crossing different varieties better, Mendel required contrasting traits in the plants. That means one character should have two contrasting traits. For example, the flower color in green peas is purple, and in some it's white. Purple and white are the contrasting traits that can help in the study. So which were the contrasting characters that Mendel found in the green pea plants? There were 7 pairs of contrasting characters in total chosen by Mendel in the plant. And here they are. The first character Mendel chose was height of the plant. The contrasting traits were tall and dwarf plants. Second character in the list was the color of the flower. The contrasting traits were purple-colored flowers and white-colored flowers. And the third character on the list was position of the flower. That could be axial or terminal. In simple words, axial means the flowers shoot off from the middle of the stem, and terminal means the flower is situated at the top of the plant stem. The next character chosen was shape of the seed. We know that green peas have 2 types of seeds. One type is round and smooth, while the other type is rough or wrinkled with scales.
I just want to say I know this part is really boring. I'm yawning as I film this. But keep in mind that Mendel must have been very bored himself. He was living in an abbey after failing to become a teacher twice. He failed the teacher exam twice. And so he was living in an abbey and his job in that abbey was to take care of the pea plants. He was in charge of the garden. And so he was probably just really bored. And if you're a bored grower of pea plants, it might be kind of fun to breed them, to see what would happen if you mixed certain plants. And so that's basically what this was. He was just bored and inquisitive, and he bred plants that had contrasting traits, which means the traits were different, just to see what their babies would look like.
This was the next pair of contrasting traits. After the seed shape, we have the character of seed color. The contrasting traits in this are the green and yellow colors of the seeds. Similar to the seed color, the next character was pod color. A pod is nothing but a case that holds the plant's seeds. Here the two contrasting traits were green colored and yellow colored pods. And the last pair of contrasting character was that of pod shape. Green pea pods can either be inflated or constricted. Both these traits are contrasting. With these 7 pairs of contrasting characters, Mendel began his experiments. Little did he know that he was building the foundation of modern genetics.
So when she says that he started doing their experiments, what that means is he would find plants that had these traits, and he would make them breed with each other. So he would make a plant with pods in this shape breed with a contrasting pod of this shape, or one with this color breed with that color, or one with this seed color breed with that seed color. So he made them breed with each other, and then he wanted to see what they would look like. What would their babies look like? That's what he was curious about.
Let's have a look at what his experiments were and the conclusions he derived from them in the next part. We have taken a look at the 7 pairs of contrasting characters chosen by Gregor Mendel for his experiments on the green pea plant. So here's a table that gives us an idea of what the contrasting characters and their respective traits were. Now, can you guess how Mendel thought of this experiment? How was it exactly planned by Mendel? What was his aim? Well,
let's talk about that actually. There are some challenges to breeding plants. You see, plants, flowering plants. Flowering plants, they are pollinators. So if we were to look at the flower, there is the female part, and we call this the ovary, and we call this the pistil. There's the female part, and then there's the male part, right? So this would be stamen, and it's easy to remember because it has has men in it. It looks like men. So there's the male part and the female part. The problem is the male part can actually self-pollinate the female part. Plants are hermaphrodites. They have a male and female part. And so they can fertilize themselves and have babies with themselves. If he's trying to breed purple flowers with white flowers, he can't allow that. He can't allow a purple flower to breed with itself. He has to make it only breed with the white flower. So how is he going to prevent self-pollination? Find out.
Mendel thought of beginning with trying to understand what happens on crossing the plants with contrasting traits. Mendel, just like others, knew that a plant with purple flowers gives us another plant with purple flowers only. And it's the same case with a plant that has white-colored flowers. Now, Mendel was curious to know what will happen if a purple-flowered plant is crossed with the white-flowered variety. Ideally, the result has to be flowers with intermediate colors. But was that actually what Mendel got? Let's have a look at the results in detail. But before that, do you know how crossing is carried out? Well, the male and the female plants are selected first. Now, the pea plant, being bisexual, has both male and female reproductive parts in the same plant. So we can choose the purple flower to be male and the white flower to be female, or vice versa. Now, the female flower on the respective plant is emasculated first. So do you know what emasculation is? As the name suggests, masculine refers to male. So here, emasculation refers to the removal of male reproductive parts. Let's take these two flowers to understand the crossing process in detail. Now, the anther of this flower will be removed. Removal of anthers makes sure that the flower...
So an anther is this part here, but the whole thing all together is called a stamen. So snip snip, goodbye male parts. What Gregor Mendel did was emasculinate one of the flowers. That's how he prevented self-pollination. And so self-pollination is no longer possible, but remember, he's in a greenhouse. There are no pollinators, there are no insects. So what he does is pretty simple. He takes, we're saying this is the male one, so he takes a paintbrush and he gathers pollen with the paintbrush as if the paintbrush was an insect, and then he simply brings the paintbrush over to the selected female and places the pollen on the pistil.
Will not get self-pollinated. Now, this exclusive female flower is dusted with the pollen grains from this male flower, which ensures pollination. And the successful fertilization will be ensured when the plant develops flowers. This is how crossing is carried out. Now, let's get back to Mendel's experiments. Mendel crossed a plant with purple flowers and a plant with white flowers.
One thing that isn't in this video that should be mentioned is that before Gregor Mendel did this, he made sure that both of these plants were purebred. So what does purebred mean? It means that all of the parents of this plant were purple and all of the parents of this plant were white for many, many generations. If you have a purebred golden retriever, it means that for many generations it has been a golden retriever. There is no other breed in the mix. The opposite of purebred for dogs, for example, is being a mutt. And so what Gregor Mendel is trying to do here is basically make a mutt, but with pea plants. And he wants to see what will happen. Which of the parents will this mutt look like?
Results in which the plants obtained will be intermediate between purple and white. But to his astonishment, he obtained all the plants in the progeny to be purple colored. Unexpected, right? And this doesn't stop here. On crossing the purple colored flowers from...
I want to pause here. So he crossed these purebreds and he made all of these mutts. Meaning each one of these is half of each parent. So he crossed the parents, we call this the P generation, to make what's called the F1 filial 1 generation. He knows that these are hybrid mutts. And so he has a question. He's curious. There is something invisible that is designing these plants. Half of it came from one parent and half of it came from the other parent. And so he called this invisible thing a gene, although he did not know it was DNA. He just called this trait a gene. What he was curious about was, are these plants here hiding the mother's white gene, or is the white gene gone? To figure this out, he allowed these plants here, these mutts, to self-pollinate with themselves, to see, is mom's genes gone or hiding?
This progeny with each other, he got another set of astonishing results. If purple flowered plants are crossed amongst each other, we should obtain which variety? We would get all plants with purple colored flowers, is what most of us would think. But to his astonishment, Mendel obtained a few plants with white colored flowers as well. So on an average, he obtained 75% plants with purple flowers and 25% plants with white flowers.
So it looks like mom's genes here, again, this is mom and this is dad. They both gave half of their traits each. It looks like we'll use W for white and we'll use B for black. Or let's just use C for color, right? We'll say capital C means purple, and we'll say small c means white. So that means that there was, each of these had a gene from each parent. And the question was, is this gene gone? Or is it just hiding? And it looks like when we allow these to cross with each other, the white gene came back. And so it was not gone. This gene never disappeared. It was only hidden, which was a big hint to how genetics works.
How could this be possible now? Let's try and understand these results in the next part.
All right, so that's part 1, and I'm going to be making the next part pretty soon, where we talk more about why Mendel got the results that he got.